Hello, everyone. I'm Kelly Ebersole, the Executive Director of the International Wheat Genome Sequencing Consortium. I want to welcome you to our webinar series. Today, we're joined by Ellie Taagen from Cornell University, who will be talking with us about positional cloning and structural variation in our most popular type of species, polyploids. So before we get to Ellie, let me give you kind of a quick overview of the IWGSC, and then we'll get started. So we are an international consortium, nonprofit consortium. We have more than 3,000 members, almost 1,000 institutes and companies that are involved with us or participate in our activities, and we're in 71 countries. We have a number of sponsors that make it possible for us to actually have both this webinar series and to keep our activities on track. So we want to thank our sponsors for their financial support. Our vision post the publication of the high quality reference sequence is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity, which is why we're very interested in Ellie's talk today. For our activities this year, we're working on establishing or expanding our collaborations with wheat genomic tool developers such as Arbor Biosciences and Illumina. We have published this year the reference sequence version 2.1 and annotation 2.1. We're in the process and hope to have by PAG a clear process for people who can submit and contribute to the manual and functional annotation of the wheat genome. And we are trying to put together funding and a project that would give us platinum quality sequences of 8 land races that would give us the full breadth of diversity in the wheat varieties. Sorry, but hit the wrong button. We're also trying to provide access for pre-publication release of genome sequences and, of course, our webinar series. So our next webinar will be on December 15th, and we'll be discussing developing sustainable strategies to protect wheat roots from take-all disease. So I encourage you to sign up for that. On your webinar dashboard, you will see a section for questions, chat, and handouts. Please submit your questions at any time during the presentation, and we will get to the questions at the end of the session. You can also monitor the chat panel to see messages from the organizers, and this might include a link to a website that is mentioned in a presentation or to a document or otherwise. You can already download the handouts of the presentations, both mine and Ellie's, in the handouts thing. Just to remind you, the webinar is recorded and will be posted on the IWGSC YouTube channel in a few days. And you can subscribe so you never miss a new upload. So without further ado, I would like to introduce Ellie Taagen. She is a PhD student at Cornell University. She is also, and has just recently been awarded, the IWGSC Catherine Fouillet Early Career Award, and she'll be receiving that in January at the Plant and Animal Genome Conference. That's a very competitive award and a competitive program, and so we congratulate her on that and wish her the best of luck in her career. So now let's hear about challenges and opportunities in positional cloning and structural variation in polyploid crops. Ellie, I'll turn it over to you now. Thank you.
Thank you so much, Kelly. Let me just get my screen set. All right, hi everyone, and thank you so much, Kelly, for that generous introduction, and to the folks at IWGSC for the invitation and the opportunity to present today, and thank you to everyone on the call for attending. Today, I'm going to present some unique challenges to positional cloning in polyploids and resulting opportunities to reevaluate best practices for successful causal variant identification and experimental design. This talk will center on a specific example from our lab, which was published earlier this year in the plant genome and highlights that redundant polyploid genomes can mask structural variation and pose a significant barrier to positional cloning in wheat. Based on what we learned, I'll present some recommendations for more robust detection of positional cloning targets, as well as how structural variants may present a unique opportunity to characterize the masked variation in polyploids. And finally, I'll introduce a repository of data and scripts that are hosted on Github that may help with the adoption of some of today's recommendations. So as Kelly just introduced, the IWGSC vision and toolbox enables wheat research communities to go beyond QTL mapping and identify causal variants driving desirable traits. And yield is one of the most important traits in a cereal crop, and it's highly polygenic in nature and is influenced by genetic and environmental signals and factors at every stage of plant growth. The more we understand about the causal variants underlying the trait balancing acts of yield, the better positioned we are to achieve genetic gains and ultimately advance towards a more integrated strategy to yield improvement. And since joining the Sorrells Lab in 2017, I've been part of the Wheat Coordinated Agricultural Project, or Wheat CAP, which is a multi-million-dollar collaboration and grant across 15 land-grant universities in the United States. And the current focus of the Wheat CAP has been to validate, characterize, and deploy the causal variants that contribute to yield components in wheat. And we've relied extensively on IWGSC annotated reference genome resources and tools for this research. So zooming in on that specific balancing act of yield improvement, grain yield is driven by a combination of yield components, which are quantitative traits themselves, such as the spikes per area, the grains per spike, and the weight per grain. And given the complexity of identifying the genetic controls of grain yield and that yield components themselves have higher heritability than yield per se, a reductionist approach that characterizes these independent traits and then their interactions is a valuable strategy to improve our understanding of the underlying genes and gene networks driving yield improvement. An additional challenge to pinpointing the genetic control of grain yield is posed by the large polyploid wheat genome, where the phenotypic consequence of a single variable locus can be masked by the redundant copies on the other homoeologous genomes. And Philippa Borrell gave a terrific IWGSC webinar last November on this subject about decoding the polyploid wheat genome. So if you're looking to learn more about managing the balancing act of quantitative trait improvement in polyploids, I highly recommend checking that webinar out as well. So how do we go beyond mapping traits that often span broad genomic regions to validating selectable markers that can drive genetic gains? Positional cloning is a foundational approach to this process, which identifies a gene through fine mapping, which involves developing a population of highly inbred plants that have a homogeneous background genome, but are segregating for your region of interest. So for example, a QTL, and then through continuous selfing, you can identify recombination events within your QTL region and more confidently associate them with the trait phenotype given the homogeneous background genome. And then exploring your fine mapping populations zoomed in region for your trait of interest with a reference genome for candidate genes, and then sequencing the candidate region and functional validation, for example, with gene editing knockouts. And this roadmap for positional cloning has been quite successful in other crops such as rice, And the introduction of an annotated wheat reference genome and the advent of gene editing methods have recently opened the door to more positional cloning opportunities in wheat. However, it is worth pointing out that wheat has some structural variation, whether that's small or large insertions, deletions, and inversions. The redundant polyploid genome can buffer those effects. So not every cultivar's genome follows the blueprint of Chinese spring, and I think that the recent focus on additional cultivar reference genomes is a very exciting area of progress for IWGSC and will continue to facilitate more robust discoveries. So now I'll introduce our case study where we hit a bump in the road for positional cloning and argue for reevaluation of how we approach experimental design for characterizing and validating causal variants. In the Sorrells lab, we have worked with a population synthetic W7984 Biopata M85 for over a decade. And consistently, we've identified a QTL associated with grain weight on chromosome 5A. There are double haploids and recombinant inbred lines and we did for this population, and we did our QTL mapping with the double haploid population because the fixed background genome made it easier to identify QTL across environments over time. And here's a graph of the QTL peaks. On the x-axis is physical map of chromosome 5A, and the greater numbers on the y-axis above the horizontal dashed line mean more significant associations between the given marker and phenotype. And the phenotypes that we're considering here as they're ordered on the plot are grain width, 1000 grain weight, heading date, and grain length. And these phenotypes were summarized as best linear unbiased prediction values, or BLUPs, so that we could consider their variation across years and environments. And as we can see on the plot, there are significant and overlapping peaks for both grain width and grain weight, as well as the previously characterized vernalization gene VERN1 associated with heading date. So I'm going to note two hypotheses that or kind of assumptions that we made here that we wanted to test, and keep those in mind throughout my talk. The first is that this is a pleiotropic QTL, and the grain width QTL peak is contributing to the grain weight variation. Secondly, we would then define the QTL by the grain weight QTL flanking markers that are on the long arm, spanning about 150 megabase pair region. So to start, we wanted to learn more about the phenotypic variation and its association with the biparental alleles in this population. And because it was a double haploid population, we could split it into just two groups with the limited genetic resolution as to whether the QTL had the OPATA or the W7984 alleles across it. And we can see that the OPATA alleles here represented in purple are contributing to the increased grain width and grain weight, but no real difference in grain length. And given the high heritability of these yield components and the significant increase in trait value in a double haploid across years and environments, we decided that this would be a good candidate for positional cloning in collaboration with the WheatCap. So in order to identify the causal variant, we developed a fine mapping population using a heterogeneous inbred family approach, or HIFS. And this relies, again, on that very homogeneous background genome that is segregating for flanking markers at your QTL. And that allows you to be more confident in associating any phenotypic variation with new recombinations across your QTL. So using the W7984 by OPATA recombinant inbred line population, we identified lines that were segregating for the flanking markers of our QTL and self them looking for recombination events that could narrow our QTL region, as well as positive and negative controls that have this very homogeneous background genome with new recombination events across the QTL. And note here, the recombinant inbred line started an F6 point, so that F6 represents the background genome, and then the red number in 1 and 5 represent the QTL region as it went from initially heterozygous to more inbred. And we pursued 5 generations of selfing heterozygous plants, screening over 5,000 progeny for recombination within the QTL flanking markers to develop our population. And here's a photo of one of the field seasons of this population development, which just requires keeping track of each individual HIF's identity for genotyping and then the post-harvest phenotype associations. And we ultimately reduced those 5,000 lines to just 109 lines that had recombination delimiting the QTL. Here, I'm just going to start with the positive and the negative controls, which have either OPATA alleles in purple or W7984 alleles in teal across this QTL region that we had identified on the long arm of chromosome 5A from 334 to 480 megabase pairs. And then to the right is the distribution of their respective 1,000 grain weight and grain width phenotypes, which for the OPATA alleles for the positive QTL variant control are associated with the greater phenotype values, and across the W7984 negative QTL control, we see that those individuals are associated with lesser phenotypic values. And then we can look at the entire fine mapping population grouped by the different recombination intervals that we identified through our fine mapping process and start a process of elimination for associating the QTL region and the phenotypes we're observing. So essentially, we're looking for a pattern where the presence of the W7984 alleles, or the teal color, is associated with a lower trait value, and the OPATA purple higher trait alleles are associated with a higher trait value. And we also validated this process of elimination with t-tests and ended up narrowing our QTL region to just 10 megabase pairs. And this region flanked 37 high confidence genes based on the IWGSC annotated reference genome. And I'll also point out that in a homogeneous background genome, we got much finer phenotypic resolution. And in the HIF population, individuals that carried the OPATA allele across the 10 megabase pair QTL were 21.3% heavier grains and 10.3% wider grains as compared to the W7984 alleles at the QTL. So in order to better understand the mechanism driving grain weight and morphology phenotypes, as well as to identify an optimum time point for a transcriptome study, we also conducted a days post-anthesis analysis of developing grains and their phenotypes. So we used positive and negative controls for the QTL HIFs in this case. And we measured grain development beginning at anthesis, so that 0 days post-anthesis just refers to 24 hours before anthesis or pollination. And then we measured the grains phenotypes at 4, 10, 16, and 22 days post-anthesis. We measured grain weight, grain length, fresh weight, and dry weight, and our days post-anthesis study had consistent associations with the phenotypic variation at 10 to 16 days post-anthesis for all traits, where the OPATA allele QTL control is significantly greater phenotype values than the W7984 control phenotypes. We also found no significant difference in the duration of grain filling among the positive and negative controls. So that suggests this phenotype is driven more by a difference in the rate of grain growth rather than the duration of grain growth. And now that we have refined our phenotype to a specific time point in development, that gave us the confidence to move forward with a transcriptome study, and that would help us refine our list of candidate genes. However, our fine mapping advancement and trait time point prioritization coincided with the publication from Gutierrez-Gonzalez et al. In 2019, where the authors pursued dense GBS linkage maps for W7984, biopotted double haploid, and recombinant redline populations. Excuse me. And one of the most relevant findings was that the short arm of the chromosome 5A for W7984's parent is missing. So this means the populations are segregating for the presence of either the OPATA short arm on chromosome 5A or the absence of a short arm. And while there were no aberrant morphological variations or infertility in our population, which are characteristic of a whole chromosome arm deletion, this report prompted us to evaluate the implications for our positional cloning efforts that we had been pursuing on the long arm of chromosome 5A. So again, although our fine mapping population indicated the QTL was on the long arm, we began by genotyping the population with 3 SSR markers that could detect presence-absence variation on chromosome arm 5AS. And you can see here, generally, more of the fine mapping population has the short arm than is missing it. But when we subset which fine mapping entries have chromosome arm short arm structural, chromosome 5A short arm structural variation, and which QTL allele they have, we see that there's only one entry where the chromosome arm is present and the W7984 alleles are across the QTL, and 2 entries where they're missing the short arm with the OPATA alleles across the QTL. So this very skewed haplotype distribution revealed that the chromosome arm structural variation and the QTL alleles are in very strong LD and had a correlation coefficient of 0.91. We also tested for an interaction between the 2 loci using a univariate linear model of 4,000 grain weight, but we did not measure any significant interaction. We can also look at this with a phenotypic distribution of the fine mapping population, where I've subset the x-axis based on the structural variant of being either absent on the left or present on the right, and then the color of the box plots and distribution of each individual line's phenotype are colored by whether they have the OPATA allele in purple or the W7984 allele in teal. And so starting with the absence variation on the left, the HIFs that broke the linkage disequilibrium and have the OPATA QTL associate more with the phenotypes that are kind of on the lower end of the spectrum per 1,000 grain weight, even though we had been considering OPATA alleles that have HIFs that have the OPATA allele across the QTL to drive a greater 1,000 grain weight. And we see that for the presence of the short arm on the right-hand side of the plot, the HIF that broke linkage disequilibrium and has the W7984 allele at the QTL has a phenotype that's of a higher 1,000 grain weight, even though we had been attributing a W7984 allele at the QTL to a lesser 1,000 grain weight. So this is a relatively small sample size, but again, very tight LD. And ultimately, this suggests the phenotypic variation could be explained by either the chromosome 5AS structural variation or the QTL alleles, despite our best efforts to develop a very homogeneous background genome in fine mapping. So as it appeared that the phenotype and the SNP correlations for the trait variation could be due to either structural variation or the QTL, we turned our attention to the transcriptome. And we hypothesized that the gene expression changes influencing phenotypic variation would have occurred before the first phenotypic significant difference in grain morphology was detectable at 10 days post-anthesis. So we sampled whole grains at 2 time points, 4 days post-anthesis and 8 days post-anthesis. And we worked with 4 different HIFs, which at this point I'll start referring to haplotypes because we have to consider the presence or absence of the short arm and which allele is at the QTL. And today I'm just going to focus on 2 haplotypes, which were the most common that we saw in our fine mapping population. One that has the short arm present with the OPATA alleles and the positive variant of the QTL, and one that has the short arm missing and the negative allele or W7984 alleles at the QTL. So here we're looking at the genome-wide comparison of gene expression for those 2 positive and negative control haplotypes. And the y-axis represents significant difference in gene expression for these 2 haplotypes. So any points on the graph that surpass the red dashed horizontal line have a significant difference in expression between the positive and negative controls. And across all HIF recombination comparisons, we identified a total of 556 differentially expressed genes, and nearly all of them are on chromosome 5A. None of the differentially expressed genes' homoeologous copies were differentially expressed. And this zoomed out view of the genome validates for us that the fine mapping population had an otherwise isogenic background and gave us more confidence to associate the phenotypic variation with chromosome 5A. And zooming in on the differential expression specifically on chromosome 5A, we see that the majority of the differential expression on the short arm is due to the presence-absence variation of that short arm. We measured 0 expression in the W7984 negative haplotype control. Highlighted in red is the 10 mega base pair region that we had fine mapped for QTL, and we detected expression from both the positive and the negative haplotype controls, but there was no differential expression between them. So these results prompted us to reevaluate the considerable resources we had invested in an otherwise reliable positional cloning approach for this QTL. It was possible that we sampled the wrong time points, or that the QTL's association with grain weight and morphology was driven by a post-transcriptional or post-translational modification. So we decided to investigate the 10 mega base pair candidate region, the raw reads that came from the transcriptome, to look for any SNP variants from the reference genome or from the samples from each other. And we only identified one variant in the negative haplotype, which has been previously identified in the serials DB axiom SNP arrays. The SNP has not previously been associated with any phenotype, and there's no literature from the Arabidopsis or rice orthologs to suggest that this gene is involved in grain development. So a different time point or post-transcriptional or post-translational modification looks a little bit less likely given the lack of coding region variation. And based on the fine mapping phenotypes that broke linkage with the QTL and the structural variant, as well as the transcriptome study, it looks like this structural variant is driving the trait variation that we see during early grain development. So while these were not our expected findings when we set out to positionally clone a QTL, it is notable that the status quo approach to fine mapping identified positive combinations of 5 independent traits, 3 of which I've touched on in this talk a little bit less: height, spikelets per spike, and grain length, and then as well as 1,000 grain weight and grain width. And these are all under independent, we hypothesize these are under independent genetic control on Opata's chromosome arm 5AS. And we wanted to explore these differentially expressed genes for candidate genes that might be contributing to the phenotypes, and we performed a gene ontology or GO-term enrichment study. Ultimately, we did not find any significant GO-terms related to our traits of interest. And this, coupled with the phenotypic variation that is characteristic more of a single gene rather than an entire chromosome arm deletion, highlights some of the challenges of identifying variation that is masked by a redundant polyploid genome. So here I'd like to shift the focus to how structural variants present a unique opportunity to identify candidate genes that are associated with yield components that we measured that are likely coming from non-additive or functionally redundant genes. In our fine mapping population, the chromosome 5A short arm structural variant can be thought of as like a large knockout study to compare to the presence of the short arm. And we sifted through GO-terms related to all of our differentially expressed genes and ended up identifying 292 with GO-terms related to spike development and early grain growth. And through literature reviews, as well as these GO-terms and searching through the orthologs of Arabidopsis and rice, we identified 9 candidate genes that are known to impact yield components in rice and Arabidopsis and may impact yield components in wheat. Ultimately, in this study that I'm presenting today, we're limited by the resolution of a knockout of an entire chromosome arm, but these genes do lay the foundation for future studies across all group 5 chromosomes to investigate the phenomenon of masked variation of a homoeolog, dosage-dependent, or non-additive and functionally redundant genes which have historically been some of the more tricky to characterize genes in polyploids and may unlock new breeding targets. So at this point, I'd like to zoom out from our case study and discuss the larger implications for positional cloning in polyploids and strategies to more wisely invest the considerable resources required for validating causal variants. A primary point that our study raises is the importance of our ability to detect structural variation. And the W7984 by Opata original crosses were made in the early 1990s, and they were two of the most widely cited and used mapping populations leading up to the advent of an annotated wheat reference genome. So aside from the phenotypic variation subtleties, something that I wondered during our study was, why did the W7984 chromosome arm deletion, why was it not detected earlier by the wider wheat community? So I'll explain with a timeline a little bit how I've explained this, or how I have come to justify this to myself. In 2011, the population was reconstructed and expanded and genotyped with SSR markers and DART markers, and those are scored on a presence-absence of genomic fragments, so it wouldn't have necessarily alerted researchers to the fact that the short arm was missing if all of the markers were presence-absence. And in 2012, GBS markers were developed for the population's genetic map, but this was again before we had a reference genome, and it may not have been obvious that they were all mapping to the long arm. The IWGSC reference genome really set the stage for our ability to identify that the short arm was missing. We first noticed in 2018 when we were doing our fine mapping population development that we are having trouble developing site-specific markers and chromosome-specific markers for the short arm using exome capture data for both the Opata and W7984 parent, given their positions on the Chinese spring physical maps. But ultimately, it was the dense GBS mapping access to a annotated reference genome for the Gutierrez-Gonzalez et al. Group that was able to detect this structural variation. It also might be worth noting that the synthetic Opata studies in the past have usually relied on a subset of either population of the double haploids or RILs, and not using the entire population might not have had a high enough frequency of the structural variant to detect the abnormality, or researchers just weren't focused on chromosome 5A. So while deletions are classically associated with aberrant morphological variation and infertility, especially an entire chromosome arm, our results demonstrate that large structural variants can go undetected for years across environments, research projects, and lab groups. And some recent studies that I'd like to point out that had heed similar caution. These authors considered 15 different bred wheat cultivar genomes, genome assemblies, and reported only 59% IBD with each other and detected extensive genomic rearrangements. So that just underscores the importance to be mindful of structural diversity in wheat, especially during positional cloning experimental design. And today I've shared our experience that approaching positional cloning based on stable and robust biparental QTLs may overlook some hurdles unique to wheat's resilient polyploid genome. And if researchers or grad students are going to continue to invest the considerable resources and time necessary for successful positional cloning in wheat or other polyploid crops, I'd like to propose some following recommendations based on our recent experience. The first would be to move from a SNP to a haplotype-based approach to identify genetic diversity. Genetic markers are often associated with a trait of interest, but the genetic marker itself is not causal. And in a fine mapping context, this can overlook linkage, especially with broad genomic regions that span the centromere, and that results in selection of false positives. So that's exactly what happened to us when we defined our QTL flanking markers to be on the long arm. Haplotype blocks can identify genetic structural variation a lot more comprehensively and precisely. In your lab, you might also be using exome or regulatory capture data. So safeguards need to be in place to detect and flag structural variation. So for example, if there aren't any genome-specific or chromosome-specific markers in a broad region, that should flag a potential structural variant and not just be blamed on a redundant polyploid genome and sometimes the challenges that we face with trying to develop a SNP right where we want one. My second recommendation is to invest in sequencing to detect structural variants. This is directly what the IWGSC supports, and it's exciting to hear about their continued progress in this field. On an individual lab-based level, though, where sequencing is probably outside of your budget, investing in longer read or greater fold coverage sequencing of your parental lines for a mapping population could become a prerequisite first step in positional cloning population development. This just really gives you a clearer picture of variants genome-wide that could be in linkage or otherwise correlated and impact your positional cloning and fine mapping population development. My third recommendation is to use the transcriptome to identify candidate genes. RNA-seq of isogenic material can identify differential expression that might be of interest to you, as well as gives you access to the coding region allelic variation. This method alerted us to variants that were of interest a lot faster than pursuing additional HIF recombinations across the QTL's 10 megabase pair candidate region. If we weren't so lucky to be working on a QTL mapping project in the W7984 biopota populations on chromosome 5A at the same time that the structural variant study was published, we probably would have pursued finer mapping recombination and just further generations and have been missing the true causal variant. Measurements across multiple time points can also open the door to developing gene network studies, and that can give you access to more candidate gene discovery as well. And then traits with broad overlapping QTLs might not be pleiotropic. I want to drive this point home. For example, in our study, we showed that grain length and grain width can both independently contribute to 1,000 grain weight. However, our hypothesis that the gene contributing to grain width was driving 1,000 grain weight variation on chromosome arm 5AL overlooked the association between grain width contributing on chromosome arm 5AS. And eventually, in an isogenic background, we identified this phenotypic variation was associated with structural variation and almost certainly due to different genes. So pleiotropic assumptions may be useful in some areas of plant breeding, but they are broad assumptions for fine mapping. And with an annotated reference genome and advancing gene editing techniques, positional cloning in wheat, I really think may become routine. But pinpointing quantitative phenotypic variation and causal genomic loci requires a more tactile approach in polyploids. Incorporating strategies that are sensitive to structural variation with the classic positional cloning population development approaches, I think can reduce the likelihood to mapping in the wrong direction and hopefully minimize roadblocks to identifying candidate genes. So just to conclude today's talk before we take questions, I'd like to acknowledge that it was a bit of a struggle to work on a project that had negative results, and especially those that implied the status quo of positional cloning, population development needed an update for polyploids. This project puzzled me for the first few years of my PhD, and it wasn't until we did the transcriptome experiment that it felt like very much made sense. And one of the ways that I managed that kind of uncertainty and trickiness with this project was creating a GitHub repository, which is a open source tool for hosting data and scripts that completely documents our paper for both reviewing purposes, but also as a learning resource and reproducibility for other polyploid researchers or grad students who might be facing similar roadblocks. This repository is hosted on the above URL, and it's referenced throughout our paper. If you go to this URL, you're taken to a web page that looks like this, that has a lot more scrollability and documentation. This is just the top of the page, and it hosts all of the data for reproducing this paper under different categories that you might be interested in. So for example, if you click on the folder that contains all of the HIF for our fine mapping analysis, you're taken to a subdirectory folder, and it contains all of the input and resulting output data, as well as the scripts for analysis. And these are written in some very user-friendly markdown formats. So even if you're new to programming, these are something that you should be able to follow along and learn from. And you can also look at the raw code with the .rmd scripts. The .md scripts are the more user-friendly version. So if we click on the .md or markdown script, we're taken to a page like this, which lists all the headers for our subsequent analysis that we took in the fine mapping population development study. For example, we can click on one of the crucial steps in the fine mapping analysis, which was when we tested for an interaction between the structural variant and the QTL. And this expands the code output for reviewing, and you can also fork this repository and learn and reproduce, as well as adapt this script for your own learning. So I imagine there are some other grad students on the call, and this was the sort of resource that really I wanted while I was struggling to decide which research questions to ask next or which analysis would be most appropriate. So please do feel free to explore this repository, adapt it, and cite it for your own work and learning purposes. And hopefully it will help with roadblocks that you might face in positional cloning. So with that, I'd like to thank the many folks who made this research possible. My advisor, Dr. Mark Sorrells, thank you for helping advise this project and the opportunity to present the work today, and to our colleagues across the lab who helped support the many years it took to conduct this project. Thank you to the WheatCap for their financial support and continuous collaborations, our genotyping partners with USDA, and of course to the IWGSC for making this research possible, providing open source tools, and the invitation to present today. And thank you to everyone on the call for your attention, and I'd be happy to answer any of your questions now.
Thank you very much, Ellie, for a really interesting presentation. And I really appreciate the fact that you are making available the Github information, because I think that should be very useful to the community and to anyone else who's actually working in wheat. All right, so let's get down to a few questions. There was a basic question early on in your presentation. What method did you use to measure grain weight and length?
Oh, good question. Yeah, so we used something called the, well, okay, so it took some time to figure out our best practice, and this is all noted in our paper. We sampled everything in replicate from our populations that we grew in hedgerows for each double haploid population, and then the HIFs, again, those are individual plants. So that's it's tricky to measure grain weight and grain morphology phenotypes on an individual plant because you have only so many spikes to work with, and there's variation across spikes. So working with many observations was useful to us across years and environments to be more confident in our phenotypic measurements, and then the best linear unbiased predictions also gave us confidence in our summary statistics to make the decisions in fine mapping. The method that we would take is to keep all of these spikes of an individual plant together, thresh those by hand to maintain the seeds because they're fragile, and that is our primary phenotype. we would take 2 samples if we had enough seeds of everything for its weight, and then on a flatbed scanner, we had a software that we purchased, which its name is escaping me at the moment, but it is in the paper. The brand of software that we purchased the scanner from, it's a Canadian company, I can tell you that. And we would just then scan the seeds to get their length and width. And the software gives you a lot of other measurements, but length and width were the ones that we saw consistently associated with weight in our population.
So just to continue on that, how did you evaluate the grain filling duration for the research?
Definitely. So that took a lot of attention to detail, starting with tracking exactly at like 0 days post-anthesis. You need to know right when that plant is going to start to pollinate. So visiting them daily, peeking in. We worked with just the middle 3 spikelets, and because those are the first to pollinate, and the most full grains. So we reduced to just ones that we knew were pollinating on a similar rate, and then would tag them with a piece of like tape that was a color that I would keep track of, meeting different days, and return to them continuously on the relevant days to know how many days that had been since they were first pollinated. And taking that all the way through anthesis, which we marked as just when the peduncle starts to turn that golden color, indicative of the grain filling ending. That's how we marked the duration from anthesis to that peduncle turning a color.
All right, great. So how did you correlate the spikelets or spike and kernel weight in your mapping population?
So we had all these phenotypic measurements, and we used RQTL for our initial QTL mapping for those correlations. And then we were able to use t-tests also based on inheritability models for mixed linear models for those correlations. We gained more confidence in them across years and environments and the homogeneous background genome. If the question is also about the association between yield components, like spikelets per spike is often at odds with the grain weight because they're competing. The spikelets per spike, if the plant has more spikelets, it can only, how big can those grains be? There's a partition of resources, again, with that yield component balancing act. And I thought it was pretty exciting that we saw a positive relationship between those yield components, the greater spikelets per spike, grain weight, length, and width. Granted, we're dealing with an entire chromosome. So this, it's going to take more resolution to break down that relationship. But yes, I was not sure that we would, especially when we were working at the QTL stage, was anticipating the spikelets per spike to the grain weight phenotypes to have a negative correlation. But I think once we, elucidated that it was a structural variant driving this, that positive relationship made a bit more sense.
Great, thank you. Is it correct that the change in grain weight and total grain weight was caused by a combination of a structural SNP on 5AL and presence-absence of one or several candidate genes on 5AS?
As could there still be causation coming from something on 5AL in our QTL region?
Right.
It's certainly possible. We're not making definitive claims that we did the necessary studies to fully rule out the QTL on a 5AL, but the few HIF lines that broke LD and their phenotypes, as well as the lack of phenotypic or transcriptome differential expression across the QTL region on 5AL, and the very small amount of just a single SNP structural or coding region allelic variation gave me enough confidence to move forward with our interest as a lab being the short arm variation, and that the causality of that single SNP in the scheme of a biparental population in upstate New York versus farmers' fields and our goals for food security and developing genes that could really make a difference and yield impact kind of is where our focus shifted. But I recognize that further studies would be necessary to fully roll out that QTL.
Great, thank you. So
if I could say one more thing about that, actually, I have a slide that talks a little bit about why did our QTL map to the chromosome A long arm, because fundamentally that's where our positional cloning difficulties began. We were on the wrong chromosome arm. And some theories that I've kind of come up with are, one, that our QTL might be insensitive to the chromosome arm presence absence because the variants are very near the centromere. And there's a tiny, like the chromosome arm deletion is not the entire chromosome arm. That little bit of centromere is required for the plant to probably maintain its fertility. So there's a tiny 13 megabase pair, well, 13 megabase pair, not that tiny, but there's a 13 megabase pair segment. None of those genes were differentially expressed, so they didn't indicate to us they were candidates. So our QTL might, with that linkage, so yeah, here's again that QTL map again of, and we have in this map a one structural variation marker representing the entire short arm. And I've just graphed it to give you the sense of that size for the physical map. The lack of phenotypic resolution for a quantitative trait in a double haploid background also can make it tricky to narrow exactly where you're mapping. And another thought I had is that the genetic map markers for chromosome 5AL might not all be genome specific. So if they're mapping to the long arm of the B or the D genome, that could be causing our peak to be on the long arm. And then finally, like our conflation of grain weight and 1,000 grain weight being pleiotropic ignored that the 1,000 grain, or that the grain, pardon me, the grain width and 1,000 grain weight may not be pleiotropic. If you take a look here, the grain width QTL is significant on the short arm, but because we decided that the two were being contributed to by the same loci, and that weight comes from width, we just stuck on the long arm because you got to make decisions at some point. But that is one of the challenges of fine mapping, is that you always have to be mindful of if you're going in the wrong direction.
Right. Well, thank you for that explanation. So one of the things I was really pleased to see is that you do have a recommendation slide. I think it's slide 20, at least on my deck, where you gave the 3 recommendations for polyploid positional cloning. And I'd like for you to talk a little bit more about what you recommend for going from the SNP to the haplotype-based approach, and perhaps even what tools you think are essential for doing that. So what can someone use? What should we as the IWGSC or the international wheat community be looking at for developing? What would be useful to help individuals make that transition?
Certainly. Yeah, so a haplotype-based approach is a little bit of a newer concept. I think it's been used in other crops, and only recently there's been some publications of it, availability of doing it in wheat, which is really exciting. And I think I can, I'm not going to find it while I'm talking, that link to the publication, but I did truly in the last, at least this month, if not this week, came out. So we know this is something that we can do in wheat. The value of it, I really think, is that it gives you a better picture of structural, like, I don't want to argue that we need to be mapping structural variants specifically. Not exactly sure what that's going to do for your breeding program or discovery, unless you want to go down the road of looking at across the homeologs and you kind of already have some built-in knockouts or knock-ins to associate with your other genomes, or especially if you're working in tetraploid wheat. The haplotype-based approach gives us a, like, it's kind of what I was saying with the SNPs being, a SNP is not causal. And we often, though, when we just have a SNP-based approach, we look across our Excel sheets maybe that we've color coded by the SNP genome to see where was there a recombination event, but that did not happen right there. You have a lot going on in between, and especially when you're working on a genetic map, that can be, so they're all just things to be mindful of, I suppose, is what I am pushing for the most. How the IWGSC and kind of future methods can support haplotype-based approaches. This just last year, the publications that I also referenced from Jemima Brinton and a large group of researchers were the
project.
Yeah, these are terrific for whether your cultivar happens to already be involved here, and maybe that's a great place to start and take advantage of these resources, or look to these publications for insights on how you can do it for your own cultivars.
So in many cases, the haplotype mapping that has been done is based on partial genome information. And given what you saw with regards to the 5AS, 5AL problem, I don't want to really call it a problem because you solved it, but does partial information is it sufficient to have partial information, or and maybe combining that with what you said for your recommendation number 2 was investing in sequencing to detect structural variants, where perhaps it's a combination where you use something that is a haplotype map or that is based on partial information, but then you need to do targeted sequencing or something. Can you elaborate on that just a little?
Yeah, definitely. I mean, I think we all recognize the struggle of like the timelines of research, the timelines of grants. You're not going to have a perfect solution unless maybe Chinese Spring is what you do all your work in. So it is a balancing act, again, of which resources you have and which ones are most important to invest in. I think if positional cloning, though, is your primary goal in your project, you do need to prioritize setting up your experimental design so that you're fully aware of the genome, especially with a quantitative trait like yield components. So in that case, like if I was to do this project again, we did have access to the exome and regulatory capture. The wheat cap funding made that possible for both our parents, which is fantastic for getting started. But even still, we weren't really mindful of the fact that structural variation could have been suggested by the fact that it was hard to make molecular markers for the short arm, because they weren't, none of those sequences were genome specific or chromosome specific. So maybe we could work towards kind of at that. I think one of probably the most accessible suggestion would be at what you could afford at a lab level before you just go forward with a biparental QTL that's doing well across populations. To be mindful of all of your chromosomes, even if something can be in linkage, something can be correlated. If you think you have a homogeneous background genome, a transcriptome study is also a great way to get a sense of that.
All right, great. Well, thank you very much, Ellie, for a very interesting presentation, and I think a very good guideline for what students can do, or not only students, but what people can do in industry to try to beef up their breeding population, so to speak. So with that, I'd like to thank everyone for their participation today, and we look forward to seeing Ellie at the IWGSC workshop at the Plant and Animal Genome Conference in January of 2022. Ellie just put a note in for that the grain phenotyping scanner is called WinSeedle. Is that correct? Yes. All right. So we'll make sure that we add that to the Q&A on the YouTube channel so that everyone can see that. So, all right. Well, thank you very much, and we look forward to seeing you at our next webinar in December and in future webinars. Have a great day.
Thank you.